Okay, a very good day to all of you all. Now this comes to the second part of the video of nutrients. Before we begin, please support this channel by subscribing to this channel, like the video, and ring the notification bell. It really means a lot. Okay, so without further ado, let's begin on fats. So for fats, what are the things you need to learn for fats? Is this that you need to understand the chemical elements in fats, understand that fats are made from glycerol and fatty acids, and to investigate for the presence of fats. What this actually meant is for investigation of the presence of fats is the experiment test. And if you look at the chemical elements, this is only mainly for students taking pure biology in O level. Okay, while the rest things that all students will actually have to know. If you take O levels biology and N level biology. So in that sense, right, the chemical elements in fats is similar to the ones in carbohydrates where you have your carbon, your hydrogen, and oxygen. Okay, but then how it is being structured is different. So if we look at the components, fats are what we call actually organic compounds that have these three elements in your carbon, your hydrogen, and oxygen atoms. Yeah, and all these can be found in the periodic table. Next is that fats actually contain much less oxygen in proportion to hydrogen. So point number two and number three are the ones that actually differentiates fats from carbohydrate. So even though if they give you a chemical formula, it has only C, H, and O. How to then from the chemical formula define to differentiate that is it a fat or is it a carbohydrate? You look at these 2 points. First of all, the fats contains much less oxygen in comparison to the proportion of hydrogen atoms. And if you recall, right, for carbohydrates, the ratio of the hydrogen to oxygen atoms is 2:1. And this is for carbohydrate. However, for fats, you don't really have this ratio. And because you don't have this ratio, for carbohydrates, there's this general formula of CmH2NON. For fats, there isn't. So fats will be CaHBOC, where A, B, and C is the respective number of the molecule. Okay, these are numbers. Of course, in exam, you do not put like alphabets per se. So in this case, right, you can see that the elements of carbon, hydrogen, and oxygen, they are not fixed in fats. That means, right, I can have 10 carbon, I can have 3, or I can have 20 hydrogen, and I can have maybe 2 oxygen. That's just I just randomly just throw out some numbers, but the key thing is you can't find a very fixed ratio for it for all kinds of fats. Okay, so what are fats made up of? Fats are actually made up of two types of molecules, glycerol and fatty acid chain. Now do take note that right, for every one fat molecule, you have one glycerol molecule, and you will have three fatty acid chain molecule. Together, these will make up one fat molecule. So one very common mistake that students have is that one fat molecule is the same as one fatty acid chain. They are two separate things. So just remember this. In terms of drawing it, do read the question because you might be required to draw this kind of general shapes in exams for the science bio students in O and N level. So what happens is so in order to hydrolyze fats, right, you need an enzyme and you need water. So like what we have seen, hydrolysis is a reaction which uses water to break down the reaction. So in this case, the enzymes that we usually use is called lipase. And so one fat molecule with water molecules will help to break down glycerol and three fatty acids. And you do see that so where do you think this structure of the H2 and O goes? It actually goes into the glycerol molecule. It actually goes to the glycerol molecule to form its component. Why? Because you see, initially there is actually an attachment here. But once you remove the fatty acids, right, at the molecular level, there's an empty space for the atoms. So that's where it is actually divided for the molecule. So maybe you have one H here, and maybe you have one more OH, OH and H, OH and H to form the molecule. But of course, it's not like this. Why? Because it is not in your syllabus to know how it actually is drawn at a molecular level. But just take it that a very simple, just a few lines, and also once like parabola shape will be enough to give you the oval shape to actually draw this structure is sufficient for exams. So there is two types of fats. We are looking at saturated fat and unsaturated fat. So what are saturated fats? Saturated fats are basically made up of fatty acid chains that are straight. And that unsaturated fats are basically fatty acid chains that has nicks. Nicks are basically your bands. And for bands, what is it and why and where are the bands? They're actually located at this warp-like portion of your chain. And you can see that for fatty acid chain in saturated fats, they're actually straight. So what causes these bands are basically the structure C double bond C, carbon carbon double bond, and then for single bond carbon bonds, that will be for saturated fats. That's why it's actually straight. So this one is also not going to be tested in your exams in why is it like this, but it's good to know. It's due to the double bond. Next is so when you combine, right, you will see that the shape is actually three straight lines with one oval shape. So it's like three French fry, whereas the other one is three curved French fries and things like this. Yeah, and most saturated fats, right, they are actually solid at room temperature. So you have to actually include the word room temperature as a point of reference. And the state is actually solid. Solid meaning like it's hardened. Whereas for the other one, for unsaturated fats, they tend to be liquid at room temperature. And it being liquid at room temperature, they belong to unsaturated fat, and that's olive oil, for example. Now, how to go about carrying the ethanol emulsion test? This is a test to investigate the presence of fats. So first, you add 2 cm cube, the magic number 2, to the food substances or food sample. Now, in the event that it's a solid food, right, you just have to add water to dissolve it. That's about all. Okay. If it's a liquid one, then it's quite straightforward. Now, in exam, right, you do know that sometimes oil, right, they actually have this you one look you will know roughly guess that that can be an oil. I mean that's actually oil. So how you look at the color. If it's a bit yellowish tinge, right? Most probably it's either egg white or it's actually fats. Chances are if it's fats, then of course fats is present, or if it's oil, fats is present. Next, you shake the contents of the tubes vigorously. Why do you have to do this? Because oil is not very soluble in a lot of things. So you have to shake it very vigorously such that the contents are able to react with one another. And then you add water, 3 cm cube of water, and then you shake again. Now, after you have shaked so many times, you decant, you can remove this water, just the water layer, but leaving whatever content is remaining. And if there is actually a cloudy white emulsion, fat is present. Otherwise, what will happen if fat is absent? If fat is absent, fat is absent. Okay, solution remains clear. It doesn't remain blue because the solution you use is never a color, so it remains clear. You also cannot say that the solution remains colorless. There's no such thing. Okay, water, I mean, yeah. So do take note of it. So what are some functions of fats? So fats can help to store energy, insulate material, solvent, etc. But more important, so what happens is, right, I'm not sure whether you have come across this diet called the ketone diet. So so in a sense that let's say you are trying to tone your body, and then you do the maybe the one punch man exercise workout, 100 sit up, 100 push up, 100 squats, and then of course the running and everything. You realize that what you're doing is you're actually growing the muscles of maybe your six pack or eight pack. Okay, but then you are not burning that layer of fats that is covering on top of the muscle packs. So even though you do about like maybe so many sit-ups, crunches, and things like this, you don't really see a very defined impact. Reason being is because the fats are still there. So even though if so you still see a very flat dummy without the defined lines. So how to go about doing it is to actually burn that layer of fats. not physically use fire and burn, but mainly you do cardio activities to cause it to actually burn off the fats. And why ketone diet then? So what happens is the nutrients group in your body that actually helps you to burn through cardio exercise like jogging and running is carbohydrates. It's only after you have used all the carbohydrates, then your body will start to use fats to burn, and that's how you lose weight. Otherwise, right, even though you keep running, you don't really see a lot of fat being burned. It's because of your diet. You are taking the more carbohydrates you take in, like maybe rice and things like this, you are actually burning those energy first before you hit the fat. So then, in that way, whatever you do, you will not see instantaneous result. You may see a bit of results, but not as much as you want it to be. So what happens is for ketone diet, right? This diet is basically having a higher proportion of fats and protein, but a smaller composition of carbohydrates. So in a way, you try to cause your body to undergo ketosis such that you utilize fats first. By burning away fats first in your body, you are actually in a way losing body fats, and that helps to gain in a tone. Later, what you do is the protein proportion helps to build your muscle mass, so it makes it more defined. But at the same time, please go and read up more about ketone diet if you are interested to try it. I am no nutritionist. I will not be liable for whatever whatever effects that you have when you want to take the ketone diet. Okay, this is on your own. But generally, these are some things in which by understanding the micronutrients and things like this, it helps you to grow. Okay. And next we look at protein. So for the protein segment, For the protein segment, again, the chemical elements in protein, this one is actually for the pure bio students in the learning objective, whereas for combined bio students in N and O level, you just have to focus on the other two. So again, same thing again, I don't really have to explain that much, but let's look at this. So you could see that for protein, right, you can see for protein, right, there have been some new elements being added. One of them is nitrogen. So what does that tell you? All your three nutrients group, you have carbon, hydrogen, and oxygen. And these are the three basic elements that you need if you ever want to survive. Okay, but of course, how it's actually structured and things like this is another case. And the introduction of nitrogen helps to give it this group. And together with all this, right, proteins, right, they're actually made up of amino acids. So what you can say is amino acids, when you have a lot of different kinds of amino acids coming together, combining together, you will then form a polypeptide, which is main polypeptide, which is also your protein molecule. Okay, so that's what it meant. So what are proteins made out of, or what do we see in one particular structure, the amino acid? So amino acid has actually three main groups. They have your amino group that is characterized by your NH2, your acidic group, which is your COOH, and then of course your R. Your R side chain is actually this very long carbon chain. So for example, right, this is how it can be done. So let's say the C group is carbon, and then you have a double. So this is the acid group. Then if you are looking at the amino group, it will be this. And then the side chain R group, the side chain R group will be a string of maybe carbon, carbon, and so on and so forth. Okay, so this is generally how they actually classify an amino acid. And if you go and Google, there is actually a table of amino acids. Um, do not worry, you don't have to memorize all this at O level. They won't ask you to draw all this in exam. I think it's an overkill. Uh, but what can happen is generally all amino acids will have these three categories. Okay, the one that differentiates between amino acid is actually the R group, yeah, the R side chain. Okay, so what are proteins then? If you look at the structure, they are the building blocks of amino acids, And that what they do is, this is important. Do remember that the name of the bonds that actually link amino acids together is known as the peptide bond. So where is the word peptide form? Here, polypeptide. So amino acids linked together by peptide bonds to actually form polypeptides, which is your protein. And this you can see, right, is maybe one continuous chain. But proteins don't work that way. Proteins will then undergo multiple folding. So from here, the straight chain will actually undergo multiple folding to form a three-dimensional molecule. So you just imagine you have one very long string that is a polypeptide chain, and then you crush it up together into maybe a string or a ball of string, and that is actually an example of a 3D molecule shape. So there are actually more things to actually know about for multiple foldings, and this one is usually covered at the A levels, where you actually learn things like secondary structure, tertiary structure, quaternary structure, and helix structure for proteins. Okay, and of course, if you want to know more, then you do a degree in life science, and most probably you will even study how these foldings work. And if you are able to understand how protein folding works, you understand the sequence of the protein, then what's stopping you from growing to develop your own protein? And what are proteins then? Proteins can be hormones, it can be enzymes. So sometimes all these kind of small little things, right, when they add together, right, allows you to build a product. So the technique of folding the proteins is like the technique of actually growing a product. And slowly you'll be able to maybe make money out of it. Like you can create your own hormone that can solve a particular disease or things like this. Okay. So next, so how do we test for protein? There is actually two main methods. Uh, the one which is you add the burette solution and things like this. Do not use that one. If you want to consider that, I believe your school would have taught you. But that is not the best way of actually writing out a solution, because there is no thing. I mean, like in exam, rarely do you see the word burette solution. Okay, usually you will see sodium hydroxide, your copper 2 sulfate solution. So what you should do is, first of all, you add 2 cm cube of sodium hydroxide to the food sample. Then you shake it. So you see the first step is to actually add sodium hydroxide in 2 cm cube. Whereas for copper 2 sulfate, you add drop by drop and you shake after every drop. So a lot of students tend to confuse which one should be first, sodium or should it be copper. So always think of it as sodium, right, starts with S, right? Maybe I have to come to school first. Okay, so S comes before C, copper. So that's some way you can remember. If not, you can have other interesting ways to remember. Then of course, then comes the observation. So observation is that there will be a violet coloration. So purple or violet solution is formed if proteins are present. Then if it's absent, if protein is absent, okay, if protein is absent, the solution remains blue. So these are some of the things that you can take notice of. And basically from here is easier for you. Okay. So then we come to the last portion of what is the function of protein. So protein is the main building blocks for your muscles and what's not. So generally you help to synthesize. Synthesize means you build new things. So this is like building. Okay, of new protoplasm. Protoplasm is mainly again the revision of your cells. It's the nucleus, the cell wall, not cell wall, cell membrane, and the cytoplasm. Okay, so if you want to understand more about cells, please go and check out my video um, on cells, on cell structure. Then you will have a better understanding. And then, of course, they will also helps for the growth and repair of worn out cells. Okay. Then you synthesize protein, enzymes, and hormones. Why enzymes and hormones? Because they are actually protein in nature. That means they are made out of protein. This one you can also check out my other videos to explain that. It's quite good. And of course, the synthesize antibodies. And antibodies are the ones that are responsible for you to fight off infection in your body. Of course, we will not go so much deep in detail what is antibodies. This one you can read up more. Okay, so once again, I have come to the end of this particular chapter. So please use these videos to complement your learning if you can, if you want. with your textbook. You still need to rely on your Marshall Cavendish textbook. That is the primary source which you want to gain more details of the information, or you can use your school notes, whichever is fine. Okay, you have your own learning style. And most importantly, please remember to support this channel by subscribing to this channel, liking this video, okay, and also, like, for example, if you are even stressed, you feel that you want to know more about bio, just slam the like button, and basically that will actually tell me that I have to create more videos. But I mean, I'm just joking. But please remember to like the videos, and if you and also click the notification bell so that you'll be informed of future videos that I'll be creating. That doesn't really involve just bio chapters, but also other kinds of content. Okay, that's all for